The discovery of linked genes and recombination due to crossing over led to one of Morgan's students, Alfred H. Sturtevant, to a method for constructing a genetic map, which is an ordered list of the genetic loci along a particular chromosome. So, if this is your chromosome, this would be the loci of the chromosome. Sturtevant hypothesized that the percentage of recombinant offspring, the recombination frequency, depended on the distance between the genes of a chromosome. So, here's a chromosome and here's another. And these would be the genes of the chromosome. And he's talking about the distance between the genes. He assumed that the crossing over is a random event, with the chance of crossing over approximately equal at all points along a chromosome. Based on these assumptions, Sturtevant predicted that the further apart two genes are, the higher the probability that a crossover will occur between them. Therefore, the higher the recombination frequency. So here are the two chromosomes, and these are the genes on each chromosome. And between each chromosome, there are multiple points. And the distance between these two genes is larger, so the percentage of the genes of this chromosome crossing over are higher because there are multiple chromosomes, or there are multiple points between the chromosome.
A linkage map is a genetic map based on the recombination frequencies, and it shows the relative locations of genes along a chromosome. A linkage map is based on the assumption that the probability of a crossover between two genetic loci is proportional to the distance separating the loci. So I'm going to go ahead and show you an example of the Drosophila gene. So here's your chromosome, and there's going to be 3 gene loci. This one is B, this is CN, and this is VG. And I'm going to show you the recombination frequencies, percentages. So right here, it is at a 17%, and that's the distance between all 3 of them. And then right here between B and CN, it's at a 9% recombination frequency. And then right here between CN and VG, it's at a 9.5% recombination frequency. So now you may be asking or saying that 9 plus 9.5 is 18.5, and the reason why the percentages are different is because in the second crossover, it cancels out the first, which makes the observed BVG recombination frequency different while contributing to the frequency between each of the closer pairs of the gene. So this is what allows this to happen.
There are some special cases where the recombination data is more complicated. When the genes are very far apart on a chromosome, the crossing over is almost certain. For example, if this is your chromosome and the genes are all the way far apart, it's almost certain that it'll cross over somewhere in between. So, though all physically they're connected on the chromosome, genetically they are unlinked. This is because they assort independently, almost like they're on different chromosomes, even though they're not. To map these, the recombination frequencies of genes in between are added together. So say we had a 15% here, and say a 20% here, and then so you had like 5% between these two, and maybe 6% between these two. All of these could be added up to get the recombination frequency of the 2 genes on the end.
Cytogenic maps of chromosomes locate genes with respect to chromosomal features such as stained light and dark bands. Using these, you can locate the position of genes using a numbering system called the International System for Cytogenic Nomenclature. The chromosomes have two arms, a short arm which extends upward from the centromere, and then a long arm which extends downward from the centromere. The chromosomes are split into regions. The first region starts at the top of the chromosome, and then it ends at the centromere. And then, so this will be region 1, and then region 2 follows that, and then region 3, and then so on. As the bands become more visible with higher magnification, each region is divided into further bands. And then those bands are divided into subbands, and these are both all represented by numbers. So the first band could be 22, and then a subband of 22 would be 22.1, 22.3, 22.2. And as the magnification gets more increased from the first band to the subband, you add a decimal. That way you can tell which is which band.
Now let's look at how we can apply genetic mapping to questions. This question is found on page 297, and it is question 3. It says, genes A, B, and C are located on the same chromosome. Test crosses show that the recombination frequency between A and B is 28% and between A and C is 12%. Can you determine the linear order of these genes? Explain. So, first what we do is we would think about how this chromosome could be set up. So I'll draw a chromosome, and we know that A and C are 12% apart, so we'll draw those ones first. A and C, and 12%. We also know that A and B are 28% apart. So, if we put B over here, then we can assume their distance is 28% apart. However, this is only assuming that the distance between C and B is less than 28%. However, if we were to draw the chromosome again, we could put C and A, in this we would flip their order this time, and once again they are 12% apart. But then we could also put B the whole way over here and say that A and B are 28% apart. And this is assuming that the distance between C and B is greater than 28%. So, since we don't know what the distance between C and B is, we cannot figure out what the linear order of these chromosomes are, although we could guess between two.